So that's our genome browser. What if we want to look at the cell's metabolism? Well, let's go back to Tools and click on Cellular Overview. Here we go. An organism's cellular overview is an at-a-glance snapshot view of that organism's metabolism as compiled from all the different reactions and pathways that are included in that pathway genome database. It also reflects a little bit of the organism's structure, at least in bacterial cases. Here you can see that we have a gram-negative organism, E. coli, and we have two membranes, and this location here actually matters. We have things that are localized to the inner membrane, things that are localized to this periplasmic space, and things that are localized to the outer membrane. And if I click over to the right here, you can see where we've stuck a bunch of the periplasmic reactions. You can also see the legend over here that tells you what things are by their shape. If something has a triangle, it's an amino acid. If something has a square, it's a carbohydrate, and so forth. Let's click back. As you'd probably intuitively guess, dots are reaction intermediates, lines are reactions. If you mouse over something, you'll get a description of what you're over. Right now, I'm over malate, obviously over the TCA cycle, and looking at the super pathway of glycolysis, pyruvate dehydrogenase, TCA, and glyoxylate bypass. Nice, huh? Anytime you're mousing over something and it gives you that symbol, you can actually click through to it. I could click through to the TCA cycle, I could click through to something else. These shadows that you see in the background here define sort of broad functional areas. You know, energy generation, biosynthesis of certain things, that sort of thing. The goal is to have the pathways stuck together in general functional groupings so that it's sort of intuitive how the whole thing is laid out. Note that if you download the Pathway Tools software, you can also generate one of these overviews in a giant full-sized wall poster. That's really useful if you've made a database for your organism of choice and you really care about the metabolism. In a later webinar series, we'll talk about how to paint high-throughput data, like metabolomics data, onto this display. Finally, in our viewing tools, we have the Genome Overview. Now, we saw the Genome Browser, where you're looking at the genome in scale, moving along through it in detail. The Genome Overview is a more schematic view of the organism's genome. It shows you all the genes at once. It's clearly not to scale, and it's really focused on where genes are relative to other genes and operon identity. So here we have operon identity indicated by color, but also indicated by lines below the operon. And this is useful because you can see, of course, there are different transcription units for the same sets of genes. You know, here we are in the CAI genes, and there's a transcription unit that just covers the first three, and then there's a transcription unit that goes all the way through. You know, so we have the option to terminate here, which is interesting. And again, later on we'll talk about viewing tools that let you put, say, gene expression data onto this display. If we mouse over a given gene, we'll see things like what the gene's name is and what its product is. And if it's part of a pathway, we'll see that too, which gives us an interesting additional view into how this gene relates to the organism's metabolism. For example, YNFH here, which is part of a pathway of NADH to dimethyl sulfoxide electron transfer. Neat. And while we're in the genome overview, if we switch to an organism with multiple genetic units, we'll see all of them. For example, let's switch over to Brucella ovis, which I presume is not so good for sheep. Now we switched to Brucella ovis, but notice the overview did not automatically change. This is what I was mentioning earlier in organism choice. This just sets up our future actions. So let's go to Tools again and pick Genome Overview again. And here we go, the Brucella ovis Genome Overview, which has both of its chromosomes. And this is what you're going to see if you have multiple chromosomes, if you have a chromosome and some plasmids. They're all going to be displayed here at once in the Genome Overview, which again is helpful because while you're in the genome browser, you actually can only look at one replicon at a time. A chromosome, the other chromosome, a plasmid, whatever. The Genome Overview gives you the full high-level overview and is useful for that reason. Okay, so now that we've looked at a high-level view, it's time to go for a little more detail. In the next part of this intro series, we're going to take a look at genes and transcription units. So please come back and look at both of those with me in the next part of the series.